Hello students, today we'll discuss the topic of structure of chromosome. Structure of chromosome. Last time I discussed about the mitosis, the division. Now we'll discuss about the structure of chromosomes. Walter Fleming he was the first German scientist who studied chromosome in the larva of salamander. So let's write here, he was the first German scientist who discovered or observed or who studied chromosomes in the larva of salamander. Salamander is an animal, is an amphibian. It is an amphibian. So actually in last videos I have discussed about chromatin and chromosomes. So you don't get confused. I am explaining the difference between chromatin and chromosomes. Actually it is a difference between these stages. In chromatin is actually a long and extremely thin, darkly stained fibers are long and extremely thin, darkly stained. Stained means darkly colored fibers. So the most important thing is when cell is not in a phase of division, when cell is not in a stage or phase of a division. So this is one of the most important thing that you should consider, that it is not in a stage of division. At that time it is called chromatin, whereas in chromosome we call it chromosomes when they are highly coiled, condensed, thick chromatin fibers. So actually they are chromatin fibers only, but when they become highly coiled, condensed, thick, then they are called as chromosomes. And it happens mainly during cell division. So these are the two differences between chromatin and chromosomes. Their chromosomes are very thin, darkly stained fibers, and it is not in the stage of division that you should consider. And chromosomes, it happens during cell division. They becomes very thick, highly thick, condensed fibers. So now going on to composition of chromosome. Let us see. Actually, chromosomes are made up of which substances? So mainly the composition of chromosomes, it is a complex of DNA and proteins. So these two are the major parts of which this chromosome is made. Let us make a flow chart to explain you clearly how this chromatin is made up of. First one is the DNA. Full form you already know, deoxyribonucleic acid. And the second one is histones. Histones are the protein only I am talking about. So DNA constitute at least 40% of the chromatin. It constitute 40% of chromatin. And the 60% of chromatin is made up of histones. Histones are the proteins. So this DNA, let's go. This DNA actually it is found in the form of a DNA strand. A strand means shaft. We can call it a rope also. You can, if you don't understand, we can call it a rope. It looks like this, like a rope. So DNA, this DNA strand is composed of three main constituents. First one is phosphate. Second is sugar. And the third one is nitrogenous base. So this DNA strand is composed of phosphate group, sugar group and nitrogenous base. These nitrogenous base are of four types, four bases. First one is called adenine, and you can also call it as adenine. It is represented by A. Second one is guanine. We can represent it by G. Third one is cytosine, that is C. And the last one is thymine, that is represented by T. We will discuss about this nitrogenous base in next video. Thanks.